Thank you very much, Dr. Adams. It's a real honor to be with you. I'm really looking forward to meet you, and at least virtually we do get to meet. So it's a real pleasure, and thank you so much for the opportunity to be here, and all the organizers of Synchrony 2020. Amazing speakers, and I'm going to give a little pediatric gastroenterology perspective of the gut part of the gut-brain axis. So the objectives would be to recognize the importance of the gut microbiome and the gut-brain axis. We heard quite a bit about the gut microbiome already. And understand ways to modify the gut microbiome to benefit the brain and benefit the gut. So just a quick definition. It's basically just a bunch of organisms living in the same space. And that space can be skin, that space can be lungs, that space can be mouth, that space can be gut. But of course, as a gut person, as a pediatric gastroenterologist, I think the gut is the most important microbiome of all. There are bacteria in the gut, not just bacteria, there are viruses and fungi. But the ones that have been studied the most are the bacteria. And there's a gamish of them. There's thousands and millions of organisms in the gut. And some we think of as good bacteria. And if you think of probiotics, those are good examples of good bacteria. So where do we get most of our probiotics? We get them from stools of healthy individuals. And some are bad. So when we see a gut microbiome that looks like there's a lot of gamma proteobacteria, for example, or a proteus, then that's usually an indication that there's quite a bit of disease happening. But then there are some bacteria that kind of sit on the fence in the middle here. They're undecided. Sometimes they are good, and sometimes they're not. So E. coli, for example, is a really good one. So there's E. coli nissl, for example. That's a type of a probiotic. It's a good bacteria. Then we all know about the bad E. coli that we hear about from the news that create a lot of problems. And as Dr. Adams mentioned, only 10% of us is actually human cells, and only 1% of us are actually human genes. We're being overpopulated by the organisms that live in our intestine. So, there's quite a bit of work that these organisms are doing all the time. Just a quick look at the milestones of the gut microbiome. We start here in the 1900s and maybe even a little bit before that with the culture-based techniques. So just the traditional thing, you have a little plate and then you're growing the organisms on the plate. There's in the 1990s, when I started my training, we had some limited molecular techniques. So we had, some PCR, very limited PCR that we were able to do. And then in 2008, the Human Microbiome Project began, and that really shed a lot of light on our understanding of what those organisms are named and what they're doing. And then 2012, the reference database for healthy human microbiome, the first reference database. And then there's the National Microbiome Initiative and a bunch of activities that are really centered around the microbiome. Why is the 1900 not good? Because most of those organisms you cannot culture, they won't grow. And many of them are very strict anaerobes, so any little bit of air will make them, will kill them. So just a quick little thing about the factors that influence the colonization. So when babies are born, the traditional thinking is they have a sterile gut. So there are no bugs at all inside mom. But then when they're born, they start to get colonized. Those organisms start to populate in the intestine. And there are quite a bit of influencing factors in how we colonize our microbiome. Some that we can control and some that we cannot control. So age, for example, I wish I can control that, but unfortunately I can't, and age really has quite a bit to do with the gut microbiome. And we even published some work suggesting that the older children, the older teenage children, are not exactly like the young adults either. They have their own distinct microbiomes. Traditionally, we thought that the first 2 years of life, the microbiome keeps changing, and then it stops at 2. It does not, it keeps changing. So I'm gonna pick just one of these factors to kind of go over real quick. And of course, it's a very, very long discussion to really discuss all of the influencing factors. But we will be talking about a little bit about probiotics, antimicrobials, and fecal transplant. So gut colonization at birth, it depends on the mode of delivery. Also depends on how big mom is, the maternal body mass index, and does it even begin in utero? We think that when the babies are born, there's nothing in their gut and there's no microbiome. But there's actually DNA derived from different organisms that can be found in the human placenta, the umbilical cord, amniotic fluid, and the meconium. So these are discoveries that we're now learning that those babies, even before they're born, they're being exposed to a microbiome that stimulates their immune system. So mode of delivery, a very fascinating area to consider. So this is a PCOA plot where, you know, if those balls that you're looking at, if they're overlapping or if they're very, very close, then similar or identical. But if they're far from each other, then they're different. So if you look here to the right, you can see that the babies that are born by C-section, so these are the light blue balls, are very, very close to the skin microbiome of mom. So when the baby is born by C-section, the first thing that the baby is exposed to is mom's skin and the microbiome of mom's skin. So you see that those babies are populated by mom's skin microbiome. On the other hand, if you look on the left here, babies that are born with vaginal delivery, those pink balls, they're very, very close to the vaginal microbiome of mom. And of course, the ones that are born vaginally are very, very different from the ones that are born by C-section. So the red is far from the blue. And of course, that translates into some disorders, especially allergies and eczema. There had not been any studies looking at autism. I think it would be a very good thing to explore whether C-section would increase change the microbiome and affect the brain-gut axis from the autism standpoint. So of course, gut microbiome people are always have weird and wonderful ideas to share. And in this particular study, they actually swabbed the babies born by C-section with mom's vaginal microflora, and they were able to change the microbiome of the baby born by C-section to look like the microbiome of a baby that's born naturally. So again, there are many factors that influence the colonization, but to show you the power of the gut microbiome, if you actually feed a skinny mouse the microbiome of an obese mouse, guess what happens to the skinny mouse? The skinny mouse becomes obese. And people thought, oh, well, maybe the obese mouse ate a little bit more. Actually, the obese mouse ate less than the skinny mouse and still gained a lot of weight. And in 2005, when this was published, that really sparked the interest and the curiosity of a lot of people, acknowledging the power of the microbiome. And just like in the normal life, there's the axis from the brain down to the glands and around the kidney that produce the different hormones. So you have the command coming up from the different parts of the brain down to the cortex and giving also feedback, allowing the hormones to be controlled. Similarly, there may be some connection between the gut and the brain, as we have heard our fantastic speakers talk about that earlier in the day. So in 1909, Cannon was the first to formulate the idea of reciprocal interaction between GI function, GI symptoms, and psychological factors. And then there's quite a bit of connections, not just one, multiple ways where the gut and the brain can talk to each other. So you have the hormones, you have the different neurotransmitters, you have the different neurological, like nerve connections, where there can be a control coming from the gut up to the brain and vice versa. So there's critical influence of the microbiome on mood and behavior. So some of these studies on mice, so these are all experimental studies where they get rid of all the microbes in the experimental animals, and they start to behave in a very different way. So those are the germ-free mice, and as you introduce the different types of organisms, you see the behaviors of these experimental animals change. Again, suggesting that there's quite a bit of a strong connection between the organisms that live in the GI tract and the brain function and the brain behavior. Similarly, the microbiome may contribute to GI symptoms by altering the gut neuromotor sensory function. So it goes both ways. It goes from the GI tract to the brain, and it goes from the brain down to the GI tract. So there are, again, many different proposed mechanisms by which the GI tract with the microbiome can produce different neurotransmitters, short chain fatty acids, affecting the vagus nerve, affecting the hormones, and ultimately affecting the mood and cognition and emotion. So, so the gut bacteria manufacture bioactive compounds that help orchestrate brain function and social development. So can the microbes modulate signaling along the gut-brain axis? So this is, as we mentioned briefly, tested in germ-free animals, looking at the dysbiosis, where the balance between the good and the bad bacteria, there's a disruption in that balance. The outcome of treating functional GI disorders by manipulating the microbiota through the different ways of modification of the gut microbiome, which is really the area of interest for my research. Diet can change the microbiome, probiotics, antibiotics, and fecal transplant. So as we all know, autism spectrum disorder, they're often associated with gastrointestinal symptoms. They may be preceded by antibiotic therapy, and we hear this from parents all the time. You know, it started when the kid had antibiotics for ear infection or different reasons for the antibiotic use, and after that you start to see the GI issues and you start to see the behavior issues. As we all know, the treatment options are suboptimal, and we're always looking for ways to contribute to the health of these young children. Families often report that diet changes and probiotics may help gastrointestinal symptoms and behavior symptoms. And for that, there had been quite a bit of interest and focus on the relationship between autism spectrum disorder and the gut microbiome. As we have heard from our stellar speakers earlier, studies have shown that children with autism spectrum disorder often have a mix of gut microbiome microbes that is distinct from that in children without the condition. And in lab animals, autism-like symptoms arise when normal species of gut bacteria are absent. And germ-free mice tend to avoid other mice. They tend to avoid new social situations and groom themselves quite a bit, quite excessively compared to the ones that have the normal gut microbiome. More proof is the gut bacteria break down food and generate products that can affect thinking and behavior. A good example of that is the Clostridia that can manufacture quite a bit of short chain fatty acids. Those are, broken down from the undigested carbohydrates that we take in our diet. And propionic acid is one of those short chain fatty acids. It's interesting to learn about the short chain fatty acids, because one of them is butyrate, for example. It's anti-inflammatory. They're regarded as helpful, in general. But propionic acid is one of the short chain fatty acids, and it's known to disrupt the production of neurotransmitters. And propionic acid can cause autism-like symptoms in rats, such as repetitive behaviors, unusual motor movements, and atypical social interactions. Mice with an autism-like condition had lower levels of bifido and blotchia in their intestine and made less tryptophan, so another metabolite, and bile acids that come from the liver and are broken down by the gut microbiome, which are necessary to produce serotonin. And serotonin is an important neurotransmitter in the brain. Children with autism have been consistently found to have lower levels of Veillon-Ricci, Corpococcus, and Prevotella. And some people with ASC could have an abnormally porous blood-brain barrier. So we have a pretty tight barrier that protects the brain. But with some individuals, that blood-brain barrier has little holes in them where some of the bacterial toxins can enter from the bloodstream and affect the brain. We also know that germ-free mice, when they were populated with gut microbes from individuals with autism spectrum disorder, the offspring of these mice socialized less, produced fewer vocal sounds, and engaged in more repetitive behavior compared to offsprings of mice that received gut microbes from donor who did not have ASD. And same thing, there are chemicals that are being produced that can affect the brain. And then mice that were given those two chemicals that were missing seem to improve their core deficit. So you actually see not just description of correlation between one thing or the other, but you actually take it away and you see problems, you give it back, and it shows improvement. So what does that all mean to us? All these experiments and all these animal studies are very interesting, but how do we translate all of this? So how do we modify the gut microbiome? So one of the things that can be done is the antimicrobial drugs. We've heard Dr. Adams talk about the study by one of my colleagues, Dr. Sandler from Chicago, who made this fascinating observation in 20 years ago, where he gave some oral vancomycin for regressive onset autism. He had 11 children with regressive onset autism, and he saw quite a bit of short-term improvement in 8 of the 10 children, so 80% of the kids. Unfortunately, this was very short-term, and after the vancomycin was stopped, and this is of course oral vancomycin, so it's not absorbed, and it does really impact the microbiome quite a bit. But once the vancomycin was stopped, unfortunately, some of these improvements were not long-lasting. So how else can we change the microbiome? We talked about vancomycin, but can we change the microbiome with probiotics? And we know that probiotics have quite a bit of a list of things that they could benefit people that are taking them. How effective are they? How long do they colonize the gut? Do they really change the microbiome? And the studies are variable. There are some studies that show that the microbiome can be changed by the probiotics, but not all the studies have shown that. And in pediatrics, we have shown that one of the important probiotic products out there really does not change the microbiome of the kids. you can see the probiotic bacteria, but it doesn't change the other bacteria in the microbiome. So this just came out a few weeks ago, the effects of probiotic supplementation on gastrointestinal, sensory, and core symptoms in autism spectrum disorder, a randomized controlled trial where they had 85 children with autism spectrum disorder. And there are some potentially positive effects of probiotics on core autism symptoms in a subset of the 85 children, independent of the specific effect of the probiotic on the GI symptoms. So some had GI symptoms, some did not. And overall, there was this was a positive study in a randomized controlled trial. So the best and the most durable way to change the microbiome is actually fecal microbial transplant. And we've heard our outstanding speakers touch on that earlier. There are many, many, many names. You're trying to hide poop, but you can't. So FMT is usually or fecal microbial transplant is usually the word du jour, if you think about it. So the microbiota can be modified through implantation of healthy microbiome, very old from Chinese healers using the yellow soup. And the first report was in 1958. So it was being done without medical reports, but the first medical report was back in 1958. Where they used it for pseudomembranous colitis, which is basically Clostridium difficile, this bad organism that people get from getting antibiotics that kill the good bacteria and allow this really bad infection to flourish. Actually, 1956, if you may remember, it was 1958, the first written report in the literature, right? But in Stanford, 2 years even before the first written report, Dr. Schachter is one of the surgeons, used to collect stools from people that are coming in for surgery. And he would actually pack those gelatin capsules with the microbiome before they go to surgery, before they get antibiotics. And after the surgery, he gives them back the healthy microbiome that they had before. So after that, there are some additional reports that looked at inflammatory bowel disease, the disorder that can be pretty severe in young people. And one of the first reports of fecal transplant was a physician who actually had the disease and self-administered the enemas and reported remission from the disease that he had been struggling with for quite some time. And then the list goes on and on and on. Just wanted to pause here for just a second. This is one of the early reports of the changes in the microbiome. So all these stacks are different organisms. So this is the patient before the fecal transplant, this first stack. And you can see a lot of blue here. And really, what's very outstanding is there's no orange. And the orange is basically Bacteroides. Those are very protective for different disorders, including C. diff. So this is the patient day 0 before the fecal transplant. You see, quite a bit of change as well. And then the donor has quite a bit of Bacteroides, all this orange stuff in the middle. And then day 14 and day 33, you still see quite a bit of durability for the fecal transplant compared to the donor, which is the one in the middle. So this was 33 days. This is going down to 128 days. These are adult data from one of my collaborators. And this is actually our data in children with C. diff. And you can see the donor microbiome, Bacteroides are the brown, and the pre-fecal transplant, you don't see any Bacteroides in this before the fecal transplant in children with C. diff. But then afterwards, you actually see it for 12 months, and you see quite a bit of durability in a fecal transplant. That's not something that you see with antibiotics. That's not something that you see with probiotics. So the most durable way to change the gut microbiome is with fecal transplant. So fecal transplant, I wanted to stop here for just a second and review safety. So in general, they are regarded as safe. The most common side effect that we see is bloating. We've done over 150 fecal transplant for children. But what we usually see, maybe about 20% of the kids will have some bloating. About 5% of the kids will have flare of inflammatory bowel disease. We are actually one of the centers around the country where we looked at safety and efficacy of fecal transplant in children. So we have about 400 children that are being followed in that cohort, and quite a bit of power. And looking at how many of these kids that have had inflammatory bowel disease in the past that had quiescent disease, so their disease was under control, but then after the fecal transplant, they flared. So that's certainly something that can be seen. Recently, last year, there was a couple of reports of multi-drug resistant organisms. So these are the superbugs that are not killed by the ordinary antibiotics, and they're resistant to several of the antibiotics. And there are actually, there was a case that got very sick, another case that died because the donor, a healthy donor, didn't know that the donor had the multi-drug resistant organisms in the poop. And the poop with the multi-drug resistant organisms was transferred with the fecal transplant and caused a lot of problems. Same thing with pathogenic E. coli and enterotoxigenic E. coli. And of course, our new player is COVID. We've heard our stellar speaker talk about sequencing for COVID. We actually screen them with PCR in the stools. So very important safety issues to consider with fecal transplant. And as we heard, the most famous study with fecal transplant on autism showing quite a bit of improvement in the GI symptoms and improvement in the autism and behavior symptoms. So an amazing study with a follow-up just from last year. So there are some clinical trials across the world. The ones that I was able to find yesterday at clinicaltrials.gov are, of course, these studies being conducted at Arizona State University, who's spearheading all the efforts there. And we also have a study at the Children's Hospital of Los Angeles looking at the effect of fecal transplant in autism. So I see that the future will be in specific microorganisms for specific children with specific disorders. And as we all know, autism spectrum disorder is a spectrum. So you may find that certain organisms may be helpful for one side of the spectrum versus the other, with GI, non-GI, and so on. So it's not all the same disorder, it's not all homogeneous. But I'm hoping that someday, maybe in my life, that there'll be the personalized medicine where we do the fecal transplant, but on a personalized basis, where you give the child what the child really needs, and none of the side effects that we went over with the fecal transplant. I'd like to acknowledge some of the funding that we've had throughout the years, and I really thank the organizers of this event, fantastic program, and I'm happy to take any questions.